Cell transport. We're going to be looking at 3 different types. The first one is going to be passive. Second one is going to be facilitated. And technically, facilitated transport could also be considered passive as well, which we'll talk about with some examples. All right, but just letting you know that ahead of time. And third one will be active transport. And if you want to know where osmosis lands in all of this, maybe you learned this from your previous grade, osmosis is still technically considered passive. Okay, before we look at passive transport, you have to remember that a cell membrane, remember the cell membrane from the organelles video in this series? Before we tackle passive transport, please remember the cell membrane. Remember, the cell membrane was one of the organelles that we looked at in the organelles video, right? In the playlist of animal systems. And it was selectively permeable, so it lets certain things to go through and doesn't allow some other things to go through. What can help determine if something can actually go through? Well, size is a big one. Shape is another. Charge is another. And solubility is another, so how good it dissolves within that medium. So what are some easy compounds or molecules that can go through? Well, oxygen is a very easy one. It can cross the cell membrane pretty easily. H2O is another. And CO2 is another one, carbon dioxide. But there are some difficult ones that find it hard to go through. So for example, very big molecules like glucose, so C6H12O6, as well as amino acids, right, the building blocks of protein, they find it very difficult to get across as well. So let's quickly tackle passive transport. So it's the movement across a semi-permeable membrane and this is key, that doesn't require energy. This is obviously pertaining to the cell, right? The cell doesn't require energy to do this movement. So there's 3 big ones that fall under this category of passive, all right? One of them is called simple. The other one is facilitated. And the last one is osmosis. Let's look at each one. So let's have this example here. Let's say we have a bunch of molecules, high concentration of them, on one side. What do you think is going to happen? It should be pretty obvious. Of course, everything goes from high concentration to low concentration to alleviate that stress, right? There's too many here. They want to go to an area where there is less clutter. So slowly but surely, this one will go here, this one will go here, this one will go here. So then that's gone, that's gone, that's gone. And eventually there's going to become an even amount. So how many are there here? We've got 2, 4, 6, 8, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, right? So we had 22 altogether. Okay, maybe I missed 1 or 2. Well, I don't think so, but you can double check on your own. But just remember that there was 22 in the beginning, right? So what's half of 22? Obviously 11. So by the end of this, if you allow this to proceed and wait a bit, there will be 11 here and there will be 11 here in order to hit an equilibrium. And because of their size or because of their solubility or because of their shape, they're able to go through easily without any added input of energy. That's why it's called simple. Very easy. All right, then we have facilitated diffusion. This is pretty much when the particles or molecules or whatever, however you want to call it, they move through the membrane with the help of protein-embedded channels. Okay, they could be channels, they could be carriers, whatever. Okay, so this one here, for example, is that carrier that's going to help these molecules to be able to go through. Now, this one actually doesn't require energy either, but it just requires that medium, that carrier, that channel on the inside to be able to help assist those molecules across. Now, the word channel and the word carrier are not exactly synonymous. Channels will help assist particular molecules to be able to go through, and carriers help assist other molecules that channels do not help and allows them to go through. So they carry different things. So I just don't want you to think that they're synonymous. But regardless, either or a channel or a carrier, they are facilitating the help of some of these molecules to be able to go through. And again, this doesn't require energy. So I just want you to remember here, it also doesn't require energy. Remember that breakup of the ATP, and that creates this explosion of energy that can be used for reactions. Okay, it doesn't require that. Again, you see how there's a high concentration here. They want to get to a lower concentration, so they still want to alleviate that concentration, alleviate that stress, want to balance that out to equilibrium. All right, now let's look at osmosis, because technically osmosis falls under the category of facilitated diffusion, because osmosis is passive. It doesn't require energy to allow those molecules to go through. But why is it facilitated? Because technically it does require those protein channels. There are some cells that use aquaporins, which are protein channels that allow things to go through here and there. So it have like these channels to allow them to go through this way or this way or this way. So there are instances where we would consider it as facilitated diffusion, but in general, we do technically consider osmosis more as passive diffusion in terms of the language and biology that we use. But the important thing for you to know is pretty much just that H2O diffuses across. That's the definition. So it's this action of specifically water going across. Which defines osmosis. Of course, it moves from an area of high concentration to low concentration. So if I were to show you like a lot of these sugar molecules just kind of floating around here in this water solution, I'll just make a little legend here. So this will be sugar, and let's say these, so maybe not so many of them, are going to be the waters. But let's say there's a lot more waters here. I'm just going to fill this whole thing with water right now. And only maybe like 3 glucose here. Let's assume that this is a semi-permeable membrane. So let's say it only allows water. Only allows water. Okay, this thin over here. So again, take a small guess. What do you think is going to happen? Pause this video. You guessed it, it's going to go in this direction. Why? Only the waters are allowed to go through this thing. But do you think these waters, right, these green ones, here, I'll just put it this way. Do you think these green waters over here are wanting to go here? Or do you think these green waters, all these guys, are going to want to go here? It wants to go to the area where it is more hypertonic of solutes. This side is more hypertonic of solutes. Hypertonic. Because there's more glucose, right? The sugars. Water tends to flow in that direction towards the hypertonic solution. Hyper means more, right? Hyper, right? Hyper means more of, a lot. This is a kind of a little trick I want you to know. We're going to be looking at 2 practice problems at the end of this video about this anyway. But it's good for you to get this right now as a preview. I want you to look at this word here, tonic. Is it hyper or hypotonic? Okay? I want you to refer that to the solute, okay? The one that's being dissolved. So obviously the sugar is the one that gets dissolved in the water, right? Water is the one that does the dissolving. So the one that's getting dissolved, the solute. Let's look at the solutes here. All these white ones, okay? All these sugars. There's a lot of them compared to these waters, right? So there's a lot more sugars. So it's hyper, there's more of these solutes. So it's hypertonic. And water always wants to go towards the hypertonic solution. And because this thing allows only water to go through, it's going to flood in that direction. It's like if you go to the movie theater or a sports game Why are the foods so salty? It's to ensure that you keep buying more and more liquids because you're creating a hypertonic environment in your body because you're just taking in so much salt. How do you alleviate that stress? You got to get water to come in to keep you at homeostasis. Otherwise, it's going to be very uncomfortable. So, lots and lots of sugar over here. We're going to solve that problem by getting all these waters to go in that direction. So something more like that. However, I want you to know that there are 3 types of these conditions. So 3 type conditions. One of them is isotonic. The second one is hypotonic. And the third one is hypertonic. I kind of already went over this though. Well, number 2 and number 3. The first type for isotonic just means that the concentration, so I'm just going to put this as concentration. This means concentration in chemistry, by the way. So the concentration of dissolved solutes inside the cell is equal to the concentration of dissolved solutes outside the cell. The concentration of the water is the same outside the cell and inside the cell too. So water will move back and forth, but it will have no effect. Why? Because it's in equilibrium. It's balanced. It's homeostatic. It's fine. So why would there be a change? So I'll just put here no overall change. Okay? So iso also means equal. All right, then we got hypotonic. This is the concentration of dissolved solutes is more on the inside compared to the outside. So you could also say that the concentration of water is less on the inside. These both stand true. Think about it. If you have more dissolved solutes on the inside, there is going to be less water. Imagine if you have 100% of something, right? Let's say 80% of it is going to be dissolved solutes. Well, it needs to amount to 100%, right? So then what's the leftover? If we have 80 for these dissolved solutes, how much water are you going to have? Obviously, it's going to be 20%. You can't go over 100%, right? So that's what this is trying to say. So again, let's do another visual here just to make it look a little bit more, just to help with understanding. Hypotonic means that there's more dissolved solutes inside the cell. To make it easier for you, like in the previous example, I just want you to think of the word solution. All right, solution. This is short for solution. If you look at our solution, that's this water here, right? The water here, the water here, the water here, the water here, that this cell on the inside, this blue thing, is submerged in. Just think of the solution. So solution is on the outside, right? This cell is swimming in it, but solution pretty much is talking about the liquid portion. Just refer this hyper or hypo prefix to the solution. If it says hypo, hypo means less, right? Hyper means more. That's obvious, right? So what's the opposite of hyper? Hypo. So instead of more, it means less. So there is going to be less of those solutes. What I just showed you a few minutes ago, I want you to think about the solutes, okay? The solutes, I want you to connect that to what I'm saying now, that they're going to be living in the solution. So if it says that there's hypo, we're going to refer that to solutes. So there's less solutes. Where? In the solution. So I might just have one over there, okay? One solute in that solution. So that would mean that this definition rings true, that there is a concentration of dissolved solutes that are high on the inside, because if there's not much on the outside, there's probably, let's say, like 4 or 5 inside. Let's do another example for hypertonic. I think you'll get it better. All right, let's look at hypertonic now. I'm going to draw that same thing. And we're going to have that cell submerged in there. Again, I want you to fixate. How are we going to write the definition? Just to look at this prefix, hyper, okay? Hyper means what? A lot, right? Or more. All right, so hyper of what? Remember, I wanted you to refer it to the solute, right? The solute, so we got more of the solutes. Where? In that solution that I just talked about. More solutes in the solution. Where's my solution? It's this, right? The water in this container. So more solutes are going to be there. So 1, 2, 3, 4, 5, 6, let's say something like that, right? So then what's going to happen for the inside of the cell? There's only going to be maybe like 1 or 2. So the definition for this one will be concentration of the solute, the dissolved solute, is less on the inside. And if you look at our image, that's exactly what we have. There's less on the inside. This is synonymous to the concentration of the water is more on the inside. Like I said, if we're looking at this at 100%, if you only have maybe 20%, okay, less, okay, the solute, okay, you only have 20% of that solute on the inside. How much water is going to be there? Well, you still have a remainder of 80%. So there is going to be more on the inside. Hopefully this tip helps. All right, important notes for cellular processes. The main purpose of eating is to what? It's to absorb nutrients. So eating is to absorb nutrients. What is included under the nutrients? Obviously amino acids, right? You get proteins, your body breaks those down, it catabolizes them into amino acids. Fatty acids, triglycerides, broken up from the fats, or nucleotides, broken up from the nucleic acids, or sugars, right, like glucose, broken up from the complex carbohydrates. To ensure that the body absorbs the max amount, so to ensure max absorption, energy can be used. To pump these, okay, to pump these macronutrients across the cell membrane. And remember, the energy comes from that ATP breaking up into the ADPs and the PIs that you're a master of now. All right, so it's very important to have that ATP floating around, because when it reacts with certain compounds in the cell, the reactions release that cell energy that can obviously be used to power all the activities that you need to do. So why are we talking about this? Obviously, you guessed it. We're looking at the second type of transport. We obviously looked at the first type of transport, passive transport. Right, so we looked at the simple, we looked at the facilitated, and osmosis can be squeezed into here too, but now we're going to be looking at the bigger one, the active transport, and this is the important one. The active transport needs this energy from this ATP. Okay, so energy is used as a pump. All right, so let's look at active right now. It moves molecules against the concentration gradient from this time low to high concentration area. Obviously using protein carriers or protein channels, but also using ATP. So using channels, and this is the important one, ATP. Here's an example of this in the small intestine. Nutrients are going to be absorbed to avoid being excreted. from your stool, right? You want to absorb as much as the food that you just had digested and churned from the enzymes and your stomach acid and the juices like from the pancreas. All of that was used to absorb the nutrients. You don't want that to be expelled out and not recirculate in your body. So in order to maximize this absorption, for example, like glucose, it has to go through the concentration gradient. And it needs to go from a low to high concentration. It's very difficult to do, so it needs ATP. It requires that ATP for that to happen. So for example, so let's say this is your cytoplasm. And the outside area here is, let's say, your intestinal space. So let's say that there's a lot of glucose. So how many is that? 2, 4, 6, 8. So let's say 8 glucose molecules on the outside, okay, in the intestinal space. But let's say there's a lot more in here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, let's say we have 10 in there inside the cytoplasm. It would be difficult for these molecules to be able to go through. You would think it wouldn't work. But in order for that to go through, you would need ATP to release that energy so that it can go through. So let's say if you're an athlete, you want to carb load, so have lots of sugars the day before your competition so that you can saturate your liver, saturate your glycogen stores in your muscles so that you have plenty of energy for the race or the competition the next day. And you want to be in tip-top shape, the glucose have got to go through. You got to saturate as much as possible. You wouldn't be able to do that in a passive manner, what you see over here, but you can with ATP. So it still allows them to go through, no problem. Another example could be the sodium and potassium pump. Let's assume that this is extracellular fluid. And this opposite side is the cytoplasm, like in the previous example. Okay, inside the cell, the jelly that's holding all the organelles. If you have NAs, ions, right, they're free floating, how are they going to get to this side from a concentration of low to high? It uses this channel. ATP is used and releases that energy so that the NAs can attach onto it and then go out to the other side. Let's say you have potassium ions floating around here, and same thing, you want to go from an area of concentration of low to high. So how are we going to get through? You need that channel. ATP is going to be used to help that. The Ks attach onto it and is able to go through. So I'm just going to simply show you the ATP. Remember, it's 3 phosphates, right? So let's say I have 1, 2, here I'll do it as a different color. I'll do it as blue maybe, alright? So you have 1 phosphate, you have a second phosphate, and you have a third phosphate for that ATP. Once it attaches to help with that energy transfer, and then K plus is able to go through, this PI must have broken off and floated away because that action over there released the energy, right? There was a big release of energy there to have just allowed that to happen. But it's okay, it's not going to be wasted. This phosphate group, once released, can bind to another ADP and then make another ATP again. The same thing happened with the previous example with the sodium. Same thing, same mechanism. Now there is something called bulk transport. Sometimes the cells need to move a large quantity of materials. So I'll just say to carry, to move large quantities of materials. Okay, in or out of the cytoplasm. There's going to be 2 main types. Both will need the energy from the ATP. So let's look at this one here first. This is going to be endocytosis. So I'll put here example endocytosis. So endo sounds like into, okay? Into. So it's going into the cytoplasm, okay? From the outside of the cell, right? So into the cell. Let's assume that this is a solid. If it's a solid, this will be called phagocytosis. Phagocytosis. also known as cell eating. Okay, so that particle gets engulfed, okay, onto that lipid bilayer. You see how a pit starts to form? Okay, so there's a pit over here, a pit. A vesicle now starts forming using that lipid bilayer medium. So you got a vesicle forming. Remember we talked about the vesicles, how they work in conjunction with one another in the organelles video? Right, it's all coming back. And then once it's released into the cell, what happens? It can get released, okay, that solid now is able to come out and get digested, right? Remember, we're on the inside of the cell right now, so this is the inside and this is the outside of the cell. And the same thing can be done for a liquid, right? If it's a liquid, we don't call it phagocytosis, we call it pinocytosis, also known as cell drinking. So it's a transport of liquids this time into the cell. And then this one here will be exocytosis. Obviously you can tell by the word exo. Sounds like exit, so it's leaving the cell. All right, so again, if this was the inside and this was the outside, it's the movement of large materials out of the cell's cytoplasm, right? So attaches to that lipid bilayer membrane medium, and same thing. It opens up, allows those particles to go through, whether they're solid or liquid. All right, so same thing, the pit started forming, the vesicle goes onto the lipid membrane, lipid bilayer membrane, breaks apart so that the particles can be digested. All right, let's look at this question here to end it off. A cell with a semi-permeable membrane, so this is your cell, right? That's the membrane on the outside, has been placed in a beaker containing substances that are dissolved in water. Obviously, the water is on the outside over here, right? The membrane is permeable to water and iodine. It is not permeable to glucose or sodium. Assume that the remainder of the solution is water. So pause the video, give this a try yourself. A, what is the percent of water in the cell? It would be 90%. How do you know? Look on the inside of the cell. It's 10%. If you want to get up to 100%, what's the remainder? 10 plus what gives you 100? Obviously 90. So 90% of water is on the inside. Remember, you have to assume that the remainder of the solution is water. We don't have any sodium in question over here, so you can just ignore kind of the sodium. And again, we've already prefaced it that the remainder of the solution is water, not iodine or anything, right? So we don't even have to look at that one either. We're just looking at water and glucose. All right, what is the percent of water outside of the cell? Again, 90 plus what is going to give you that 100%? 10. So obviously it's 10. B is 10. Will osmosis occur? Yes, it will. Because you have so much water in here, wanting to ensure that there is homeostasis, so it's going to go out where all the glucoses are to alleviate that excessive amount of glucose. So in what direction will the osmosis occur? Obviously out of the cell. So out of the cell. Will glucose diffuse? No. Remember, the question says it is not permeable to glucose, so only water is going to be moving, so this is absolutely just osmosis anyway. So answer is no. F, will the cell shrink or swell? Obviously it's going to shrink. Because if all the water gets out, it's going to shrivel up and this thing's going to get smaller. It's lost all that volume of water. How do you know? Because obviously the water, like I said, has to alleviate that stress of high concentration of glucose on the outside. So I'll say to achieve equilibrium. All right, so the diagram shows the cell in what kind of solution? Hypo, hyper, or iso? Remember, focus on the solute with these prefixes, hypo, hyper, iso, and refer it to what it's living in. It's living in the solution. So where is the solution? It's on the outside over there. Is a lot of this solute in the solution? Yes, there is 90% glucose being dissolved by the water. This is obviously the solute. And 90% is a lot. So this is hyper. It's hyper. A lot of solute is living in that solution. So that would be the answer.